So let's now review chromosome structure. We will step back a little bit to chapter 2 and review the chromosome. We know that one chromosome can consist of one chromatid or two chromatid. So when do you find chromosome which is composed of one chromatid, meaning one DNA molecule? You remember cell cycle in G1 phase. After S phase, during S phase, the DNA is replicated, and towards the end of S phase and during G2 phase, you will find in cells chromosomes which consist of two sister chromatids. Two sister chromatids equal two DNA molecule, two identical molecule of DNA. So this is just recall what you learned before. So now, in order for a chromosome to be functional, fully functional, it has to contain centromere, telomere, and origin of replication. If origin of replication is mutated, DNA is not able to replicate, and you can imagine outcome of that. Now, what is centromere? Centromere is that constricted area within the chromosome where kinetochore attaches to. You remember kinetochore is complex of proteins which interact with the region of the chromosome in centromere and which attract and interact with spindle microtubules. Now telomeres you find at the end of each DNA molecule, and telomere protects the end of chromosome which are normally sticky. If you eliminate telomeres, the chromosome will interact with each other, will aggregate, and will actually lose the functionality. So let's now discuss in a little more detail centromere and telomere structure. So recall, centromere is that constricted region of chromosome. It does consist of tandems of short DNA sequences of tandem repeats of adenine and thymine. Actually, adenine and thymine for 90% of the centromere. Here is the sequence outline. Most of centromeres are made of heterochromatin and they do not carry any functional genes. Surprisingly, no specific sequence that would be found in all centromeres was determined. So how is centromere defined? So research suggests that centromere are not really defined by DNA sequence, but rather it's defined by epigenetic changes of chromatin structure. And nucleosome in the centromere of most eukaryote actually possess a variant of the histone. They do not have in this region histone H3, but instead there is a variant SEN H3, which might bring about changes in the nucleosome and chromatin structure. Now, we already talked about the importance of centromere for kinetochore attachment. Centromere has another additional important function. It helps to control cell cycle. Actually, centromere inhibits proceeding from metaphase to anaphase until the chromosome is properly attached to spindle fiber. So it sends inhibitory signal until all centromeres are properly attached. So the importance of centromere is actually outlined in the following experiment. The importance of centromere in the proper movement of chromosome has been recognized very early by the geneticists, and what they observed that if the chromosome undergoes chromosome breaks right here, in mitosis, fragment which do not have centromere do not attach to spindle fiber and do not move into the poles. So these fragments will be lost after mitosis and basically they will degrade. So remember that what is important here, centromere is a place where kinetochore attached, and it has been suggested that that SEN-H3 histone, which replaces H3 histone, bridges changes or brings changes in the nucleosome and chromatin structure, which promotes the formation of the kinetochore, and kinetochore is necessary for the attachment of the spindle fiber to the chromosome. The telomere structure is always found at the end of chromosome, so it's at the end of DNA molecule. And what is the function? It is a cap stabilizing function. Telomere have similar function as a cap on the shoelaces. If you still wear shoelaces, you know what happens when you pull out, you break the cap, so shoelace degrades. What happened when you break the chromosome? It has actually been shown by Herman Miller and Barbara McClintock a long time ago that broken ends of the chromosome produce unstable ends that are very sticky, so they tend to stick together and trigger degradation of the chromosomes. So end of chromosome are protected by this cap formed by telomeric sequences. They are found in all kinds of eukaryotes, in protozoan, plants, human, and the sequence, so-called telomeric sequences, are very similar in all of them. They contain several adenine or thymines, which are followed by cytosine and guanine. In humans, the telomeric sequence is outlined right here. So it is from 5' strand, it is here, right here, TTAGGG. If you read second strand from 5' and it's CCCTAA. The string of cytosine and guanine tends to be always oriented towards the end of chromosome, and the G-rich strand right here always protrudes beyond the complementary C-rich strand, and this protrusion is found on 3' end of the chromosome, and it is called G-rich single-stranded overhang. Very often this overhang can form T-loop right here outlined. However, it is not the T-loop which protects the end of chromosome. It is a multiprotein complex which is called shelterin. Shelterin binds to G-rich single-stranded sequences in the telomere and protects the end of the chromosome. So let's now very briefly review DNA sequences that you can find in eukaryotic chromosome. There are two major groups: unique sequences and repetitive DNA sequences. Unique sequences of DNA exist either as a single sequence, or you can find only few copies of the same sequence throughout the genome. So unique sequences are those which encode functional proteins. Sometimes you can find similar but not identical sequences of DNA, and these sequences actually arose by duplication of already existing gene, duplication and rearrangements, and we call this kind of DNA sequences of gene families. You know, gene family, let's say genes for immunoglobulin gene families. You know that individual immunoglobulins are different, but they share very high sequence homology. We have cytokine gene family, we have chemokine gene family. They are each different, but they share high similarity. So those are gene families. Gene families encodes functional protein. They are similar but not identical. We can find in our genome two major groups of repetitive DNA, either moderately repetitive DNA sequences or highly repetitive DNA sequences. The length of moderately repetitive DNA sequences usually varies between 150 and 300 base pair. Now, they can exist either in tandems, as a tandem repeats, or as interspersed repeat. Tandem repeats actually appears one after another, and they are usually clustered at particular location of the chromosomes, and you have two examples of those tandem repeats, transfer RNA and ribosomal RNA. Contrary to tandem repeats, interspersed repeats are interspersed throughout the whole genome. They can be either short or long interspersed elements, the signs or lines. The example of sign is alu element. Alu sequence is very interesting sequence because it is kind of exceptional. It is present in more than 1 million times and comprises approximately 11% of human genome. Also, it has no obvious cellular function. ALU is actually transposome which has been inactivated throughout the evolution. It does not transpose, and it can still serve us. For example, if you work with viruses such as HIV, which integrates into genome randomly, and you want to find out whether in your cells a virus is integrated, you can actually design PCR primers where one will be from the virus and another will be from ALU sequence. There is high probability that at least in some cells the virus will integrate in the close proximity of ALU sequence. So let's move on and talk about lines. Long interspersed elements or sequences are very similar structurally to retroviruses, and they are predominantly retrotransposomes. Now, second group of the repetitive DNA, highly repetitive DNA, is formed by sequences which are short. They are usually less than 10 base pair long and exist in hundreds of thousands up to millions copies repeated in tandem and cluster in certain region of the chromosomes. And we already discussed two of them. We discussed centromere and telomeres. Oops, there is a typo. Sorry. Now, very often these highly repetitive DNAs are referred to as a microsatellite DNA. Why it is? This percentage of these 4 bases which repeats in tandems, or 2 bases which repeats in tandems, are highly different from the surrounding DNA sequences, and therefore when you do DNA separation by combination of the high speed and density gradient, you will find that they separate as a satellite. They do not separate with a whole DNA. So it is because of the composition. Highly repetitive DNAs, similarly as we discussed in centromere and telomere, are rarely transcribed into the RNA. Now, the functional role of these DNA sequences, these repetitive DNA sequences, was not really clear, and recently it has been addressed, and it has been established that actually most of the genome, even the sequences in this highly repetitive DNA areas, are transcribed, that at least 80% of the sequences of the DNA are transcribed. And many of them actually might help or be involved in the control of gene expression.